All right. Well, welcome to all who are here with us today. Thank you so much for joining us. My name is Mark Oldham, and I'm the treasurer of the American Delirium Society. On behalf of the ADS, American Delirium Society, I'd like to welcome you to our webinar entitled Delirium Neurophysiology, Moving from Theory to No-Brainer. And so it's no secret that neurologists have historically been underrepresented in the delirium field. And we at ADS would like to see that change. And so you'll notice today, our speakers are both from neurology. And so we're honored to have Dr. LaHue, neurohospitalist from UCSF, as our presenter today, and Dr. Rabenstein, a neurointensivist here from Mayo Clinic. And they're going to be presenting delirium from a neurologist's perspective. And I think you guys are really going to enjoy this. You guys are in for a treat. We'd also like to thank several different folks. First, we'd like to thank our joint provider, Sutter Health, for offering interprofessional continuing education for this event. Next, we'd like to thank Tully Vision for hosting this event. And then a special thanks to Johns Hopkins Geriatric Workforce Enhancement Program for partnering with ADS and sponsoring the event today. So on the next slide here, in terms of let's see here. Could we go to the next slide, please? Oh, perfect. So on this next slide, in terms of CME information, Sutter Health designates this live activity for an hour of credit for physicians, nurses, and pharmacists. For professionals in other disciplines joining us today, we would encourage you to check with the regulatory board for your discipline to confirm what form or what type of credits meet your CE requirements. And then finally, here on our last slide, in terms of disclosures, our presenter, Sarah Hugh, reports no relevant disclosures. And although our moderator, Dr. Rabenstein, reports the relationships listed here, none of these were found to be relevant to this activity. And no one else involved in the planning or presentation here today reports any relevant disclosures. And so still on this slide, well, actually, yeah, we can let's get started here in a second. Apologize. So finally, two housekeeping tips to note. Number one, at the end of this Q&A, we'll be providing a URL link and a QR code if you'd like to claim CE for today's event. And also, all attendees will be receiving an email in the next few days inviting you to claim credit if you haven't already done so. And number two, importantly, please use the Q&A feature. So given this the webinar, we don't have the chat enabled, but please use the Q&A feature. Use it early, use it often. The earlier you enter your questions, the more time it gives our presenter and moderator to review and to begin developing responses to your questions. So again, we'll have the CME information at the end, but without further ado, I'd like to welcome our presentation today. Thanks guys for joining us.
Hi, everyone. I'm Dr. Sarah LaHue, and I'm an assistant professor in the UCSF Department of Neurology. I'm delighted to discuss delirium neurophysiology, moving from theory to no-brainer. This is a photograph of a brain that I took at UC Berkeley. I was a molecular and cell biology major with a neurobiology emphasis. This was a hands-on seminar led by the late Professor Marion Diamond, one of the founders of modern neuroscience, and one of my inspirations for becoming a neurologist. I'm specifically a neurohospitalist, which means I'm a neurologist who practices in the hospital. And because of this, I see many patients with delirium. And so this relates to the career advice that I always go back to, which is that you should study what makes you mad. And so that is what I'm doing. My research has spanned methodologies in molecular biology, to studying functional networks with EEG and fMRI, which is helping to inform this discussion. So to provide an outline from DNA to networks, first I'll provide some historical context. Then I'll discuss the tools we have available to make scientific inferences. I will then discuss how delirium risk factors are influenced by neurobiology and how they're informed by neurobiology. I'll do this by reviewing cells and intracellular communication, as well as the lymphatic system, blood vessels, and the blood-brain barrier. I'll then discuss how functional networks connect to all of this. And the primary undercurrent of this discussion will be what happens when things go wrong, and how does that relate to delirium? So my goal here is to provide a framework for delirium rooted in neurobiology, rather than focus on single studies in particular, though these are referenced on the slides. And note that due to the large number of references as the basis for this discussion, I'll only include key ones on the slides, but please contact me for a more comprehensive bibliography if desired. So where have we come when it comes to delirium? Well, this would not be a neurology talk without a history lesson and a story about the importance of language that we use as delirium researchers. So delirium is one of the first mental disorders described in the ancient literature. Hippocrates detailed a syndrome of acute fluctuating confusion called phrenitis. It was described as mental abnormalities caused by fever, poisoning, or head trauma. And then the Roman philosopher Celsus coined the term delirium. And so in his writings, he recommended wine for non-febrile causes and a quiet environment for sleep. He also observed that some people recovered from delirium, but some also had lasting cognitive side effects, which is what we see today. And I always find it humbling to think how similar these recommendations are to what we still do in our practice now. So one of the benefits and challenges in studying delirium is that delirium affects all patients, young and old, those with medical comorbidities or surgical indications, people who are hospitalized and those who are not. And this underscores the wide impact delirium has on our communities, as well as the urgency with which we need to advance our understanding of its pathophysiology. However, the terminology we use is varied, and in some ways that can get in the way. So common terms include acute confusional state, altered mental status, postoperative confusion, acute brain failure, encephalopathy, ICU psychosis, and countless others. Terminology can also vary by specialty. So for instance, in inpatient neurology, which is my subspecialty, terms like altered mental status and encephalopathy are pretty common. But in critical care, acute brain failure may be leveraged instead. And how we view the world is largely contingent on the language that we use. And so these differences are important. Just as there are a few neurologists studying delirium compared with other subspecialties, neurologists are not always included in the conversation. This is because, and this became most apparent to me when I joined the American Geriatric Society. This was when I filled out the membership form, and I had to choose other as my subspecialty because neurology was not among the 29 options. So there are many challenges with moving the needle on delirium prevention and treatment. There can be inconsistent approaches to delirium diagnosis. There's also the risk of delirium misdiagnosis. There are challenges with interventions due to delirium having multifactorial etiology, as well as occurring in a variety of settings. And then the fact that delirium pathogenesis remains elusive. So approaching delirium through a neurobiology lens is one way neurologists may help to advance the field. And so my goal here is to provide a framework rooted in neurobiology in order to illustrate theories and how those theories have been put into practice. So we have many tools that we can use to study delirium in terms of neurophysiology. Of course, we have blood, and we have many markers in blood that we'll discuss, and that includes studying DNA as well as RNA. We have tissue that we can look at with various types of imaging modalities. We have cerebrospinal fluid, which is my favorite type of fluid as a neurologist. And then there are going to be many, many more tools down the pipeline. And how exactly are we using these tools to study delirium? And that's going to be one of the main areas of discussion here. So this is the first MRI scan of the brain. You can see that this was a huge accomplishment, but we have made tremendous strides in terms of our ability to actually image the brain. And that strength continues to grow over time. So initially we have 1.5 Tesla scan on the left side, all the way up to a 7 Tesla scan. And really the boundaries of that are being pushed even higher. And with that, the resolution increases dramatically. And so this is, I think, a great example of how we're really only as good as our tools when trying to investigate delirium and some of the underlying pathophysiology. Some of the more common tools are also EEG, looking at EEG in terms of its frequency bands, which we have here, and then of course fMRI and NEG, and many other types of tools that I'm not even discussing here. But these here are important for studying functional networks, which I'll get to towards the end. So moving into neurophysiology, really going from cells to networks, it's important to start from the beginning and just give an overview of the flow of information from DNA to protein. So here I have an illustration of your basic cell, and you have DNA that's transcribed into messenger RNA. And then once in the cytoplasm of the cell, the messenger RNA can be used to construct a protein. And there have been many studies on the genetic associations with delirium. Many of these looking at cognitive resilience and neurodegeneration, with ApoE4, for example, is one of the more common genes of study. KIBRA and FKBP5 is from some recent work of mine that looks at genes that confer cognitive resilience. There are also hormones and neurotransmitters that are studied. So looking at melatonin receptors, and COMT, which is an indication of neurotransmitter metabolism. And I think it's important to note that gene expression differences are really things that need to be studied in a diverse population. And I think this is an area of study that we need to think about critically as delirium researchers to make this one of our goals. Taking this one step further, we also have RNA sequencing, and this is something that has been studied in just small populations of patients with delirium. But I think this is an important background with which to give this discussion, because we do see these hints of there being significant activation of inflammatory signaling molecules such as cytokines, the complement system, which is also implicated in inflammation, and others. This is giving us some of the foundational work for thinking about inflammation and delirium, which I'll talk about more in detail. So to contextualize the location of some of these basic cells, we'll talk about the brain later, but here you get a little bit of a preview. We have the cerebral cortex here. You have the gray matter, which is where the cell bodies are located. The white matter, which is where the relay of these cells is formed. And so from that, then you have the cell body up at the top and the axons that form this communication network. And so to talk about this neuron cell in more detail, there are approximately 10 to the 12 neurons in the brain with 10 to the 15 synapses. The way this starts is that you have the dendrite, which is up on the top left. This is what receives the impulses from the synapse and the synapse of the adjoining neuron. And this is what carries the impulses to the nerve cell in the nucleus. And then this travels along the axon, which is that longer branch there. And this is really what carries the electrical impulses from that cell body all the way down to the synapse. And the synapse then is a structure that allows the neuron to then pass these electrical and chemical signals to other neurons or other cells nearby. One of the tools that we have is looking at markers of neuronal damage that can be measured largely in spinal fluid and blood. These are markers that are associated with numerous neurologic disorders. So yes, they're studied in delirium, but really these are important markers to study in a wide variety of neurologic conditions. Neurofilament light chain has received significant attention. This is a marker that specifically looks at axonal injury and degeneration. And this has been shown to be increased in CSF and serum, and that's been associated with delirium in addition to cognitive decline. And then there's also neuron-specific amylase, which is a marker that's increased in plasma that's been associated with delirium as well as mortality. And this is also something that we use for neuroprognostication after cardiac arrest. So you can see that this has wide-ranging effects. So neurodegenerative markers are a common area of study in delirium. Still not clear if these serve as risk factors or distinct pathophysiology for delirium. Tau is one that stabilizes neuronal microtubules. It's been studied in association with delirium severity, where the increase in spinal fluid tau is associated with that. And also changes in plasma tau following surgery may be associated with delirium. Amyloid beta is also a key area of study. The function of amyloid beta is still not entirely clear. It may protect against oxidative stress. It may also help to regulate cholesterol, which is an important pathway when thinking about neurodegenerative diseases like Alzheimer's disease. And this is something that is derived from amyloid precursor protein in a cholesterol-dependent process. And so that's something that is regulated by ApoE. And so low cerebral spinal fluid amyloid beta is something that is associated with delirium, though the direction of this association is not always consistent. Oftentimes when we're looking for amyloid beta in spinal fluid in people with dementia, we tend to see that it's lower, largely because this is something that aggregates. And so the actual amount of it that is circulating tends to be lower. Next, I'll talk about glial cells. This is a way that we can move beyond this neuron-centric view of delirium. And these are important cells when thinking about numerous neurologic diseases and are becoming more and more important in the study of delirium. First, I'll start with the astrocytes. Astrocytes are really the workhorse of the CNS. These outnumber neurons in the human brain. They play a key role in numerous functions within the central nervous system, and these include water homeostasis, defense against oxidative stress, energy storage, helping with mitochondrial health, tissue repair, and also blood-brain barrier permeability, which is going to be a key factor that we'll discuss later on. Next, we have oligodendrocytes. These are the myelinating glia of the central nervous system. The myelination of the axons of neurons allows for the rapid conduction of these nerve impulses and contributes to the integrity of the axon. So this is one way that glial cells are very directly helping out with the support of neurons, and here they're key for communication. Next, we have microglia. These glial cells mainly help to mediate immune responses in the central nervous system. They act as macrophages. They help to clear cellular debris and dead neurons from nervous tissue. And then lastly, we have the ependymal cells. The ependymal cells are very important for homeostasis of cerebral spinal fluid and are also important for the generation of spinal fluid. These, in addition to the other types of glial cells, further help with waste clearance. This is another illustration of an ependymal cell, which you can see here that was taken with an electron microscope. The ependymal cells are at the top there. You can see these are column-like cells. The spinal fluid is there at the bottom. You can see this direct relationship with the spinal fluid. Essentially, they're ciliated epithelial cells, and these are largely located along the ventricles of the brain and the spinal fluid. Again, they play a critical role in cerebral spinal fluid homeostasis and clearance of waste products from the brain. So how do we become glial inclusive? So microglia and astrocytes become activated with increasing age, neurodegenerative diseases, and inflammation. Glial activation is also called priming. And this is important because it can lead to the production of pro-inflammatory cytokines and reactive oxidative oxygen species. And once these glial cells are primed, they actually, after having subsequent exposure to provocation, there can be further activation that leads to increased amounts of these factors. This is also important for the creation of tau neurofibrillary tangles. Next, we also have cytokines, especially things like interleukin-1 beta and tumor necrosis factor. All of these are key for thinking about inflammation of the brain as well as systemic inflammation. And a key role of cytokines is to inhibit acetylcholine release in the brain, which is important for cholinergic dependent memory function. And so next I'll talk about markers that are primarily expressed on astrocytes that are key measurements in some biomarker studies in delirium. S100B is one of the more frequently cited ones. This is widely expressed with varied intra and extracellular functions. It's released from cells in the setting of tissue and cell damage. And some studies have shown that higher levels of S100B are seen in people after developing delirium, especially in those with pre-existing cognitive impairment and also those with infection. Next, we have soluble triggering receptor expressed on myeloid cells too, which thankfully has an acronym. This is an innate immune receptor that's expressed by microglia specifically. This is something that is studied in neurodegeneration, generally speaking. It's been seen to be elevated even in prodromal Alzheimer's disease, and also has been shown to be found in higher levels in spinal fluid in those older adults with delirium. There are additional markers that are primarily expressed by astrocytes. Glial fibrillary acidic protein, or GFAP, is one that's expressed by astrocytes. Increased GFAP indicates astrocyte activation, and that can correlate with cell damage and death. In one study of postmortem tissue in adults with delirium, increased activity of GFAP was found. However, plasma studies looking at GFAP in people with delirium have been less consistent. The last one I'll talk about is matrix metalloprotease 9, or MMP9. This is also expressed with astrocytes. This is something that could be an important regulator for amyloid beta metabolism, and also play a role in this astrocyte-mediated amyloid beta degradation. Lower plasma MMP9 is a marker that's been associated with ICU delirium. Next, I'll talk about the communication between neurons and glial cells. It's important to understand that neurons and glial cells work together. These are not distinct entities. This is going back to the fact that these oligodendrocytes are intimately connected with neuronal function, but also this communication between neurons and glial cells is key even just for the functioning of metabolism within these cells. So one example is that these cells communicate with different neurotransmitters. You can see on the left, there's a neuron that represents an excitatory neuron using glutamate, which is an excitatory neurotransmitter. On the right, there's a neuron cell that is an inhibitory neuron, which uses GABA as a neurotransmitter. And both types of cells interact with astrocytes, and it's very much a bidirectional relationship. These transmitters, both neurotransmitters as well as gliotransmitters, include glutamate. And this is key because the fact that glutamate is one area that these are connected, having too much glutamate as well as too little glutamate can result in pathology of the cell. As you can imagine, that if there's increased excitation, that is something that could also damage the cell. One of the most important relationships here is the generation of ATP. And so things like hypoxia and inflammation, these can result in mitochondrial dysfunction within the cells, and that can result in impaired oxidative phosphorylation, leading to insufficient ATP, and so reductions in energy that's available to these cells that ultimately results in metabolic and energy dysregulation. Lastly, the primary way that these cells communicate is another glial transmitter, which actually are cytokines, such as tumor necrosis factor, which is something that is secreted by these astrocytes and is an important transmitter that's expressed by these cells. Next, there's also intracellular communication, more broadly speaking, largely through neurotransmitters and hormone metabolism. Acetylcholine dysregulation is one of the more commonly studied neurotransmitters, both when thinking about cognitive function as well as delirium, broadly speaking. This neurotransmitter is key for maintenance of alertness, attention, learning, and memory. It's also implicated in anti-inflammatory pathways. So this is a neurotransmitter that's released during tissue damage or infection, and is something that can help inhibit cytokines. Anticholinergic medications can increase the risk for delirium. And so this is one of the more indirect signals that we have that this is an important pathway for delirium pathogenesis. And the use of acetylcholine has also been measured in fluid from delirious patients, including in spinal fluid as well as plasma. And low levels of acetylcholine in these fluids has been associated with delirious patients. This specifically has been studied in ICU populations. Lastly, we know that acetylcholinesterase inhibitors, which inhibit the breakdown of acetylcholine, are helpful for several types of neurodegenerative diseases. So thinking about Alzheimer's disease, Parkinson's disease, especially dementia that's associated with Parkinson's disease. And there have been variable benefits in delirium. So moving along with intracellular communication, tryptophan dysregulation and tyrosine dysregulation are which are precursors to serotonin and catecholamines like dopamine, norepinephrine, have been studied in delirium as well. Again, there was a study looking at critically ill patients where concentrations of plasma tryptophan and tyrosine were associated with this transition to delirium in mechanically ventilated patients. And so this suggests that alterations of amino acids may be important in the pathogenesis of delirium. And just thinking about some of these upstream pathways of the creation of these neurotransmitters, and also things that may be a little bit easier to study than neurotransmitters themselves. Importantly, we also see changes in plasma esterases. And these esterases are things that affect neurotransmitter function. They can break apart the neurotransmitters. And this is also something that can be implicated in drug metabolism, which is an important area of study for medications that are used either for delirium prevention, though really more so for thinking about risk factors for delirium. So one way that neurotransmitters interact with each other is through this very important ascending reticular activating system, or RAS. What this is a network of neurons that are located in the brainstem that then project to other regions of the brain using different types of neurotransmitters, as indicated here, things like serotonin, dopamine, acetylcholine, and they project to these other regions of the brain in order to mediate both alertness and behavior. This is the schematic to show the different layers of protection for the brain as we work our way outward from cells and then looking at tissues more broadly. Here are the brain parenchymas in beige. You see the cortex there highlighted. Then above that, you have the subarachnoid space, and this is where you'll find blood vessels and also cerebral spinal fluid. And then above that, you will then see the skull as well as the scalp most externally. So I'm going to spend a little bit of time on the lymphatic system, which is essentially the glial lymphatic system. This is the central nervous system, a form of a lymphatic system. This was only first described in 2013, but serves really a crucial number of functions for the central nervous system. What this is a system that aids in the transport of nutrients as well as signaling molecules into the brain. And it also promotes the clearance of proteins and waste products out of the brain, including things like amyloid beta, which was implicated in neurodegenerative disease. And so what you see here on the left side, you have artery in the red, and then on the right side, you have a vein in blue. And what you have is spinal fluid that enters into the brain through these para-arterial spaces on the left side, which then mixes with interstitial fluid and waste from the brain parenchyma. This then drains through the paravenous vessels through actually the astrocytes. So this is ultimately why it's called the glial lymphatic system, because it really depends on the function of glial cells. Here, the green arrows in shades indicate the spinal fluid and interstitial fluid transport from the left side of the screen to the right side of the screen. And then the black stars here indicate interstitial waste products that then exit. Zooming in a little bit, as we go from our lymphatic system, and then zooming in, we'll take a look at one of the vessels on the bottom left side of the screen. This is called the neurovascular unit, and this is at the level of the intraparenchymal capillaries themselves. And what this is that it includes these perivascular astrocytes. It also includes neurons, microglia, and endothelial cells that line the vessel. And these capillary endothelial cells, along with the astrocytes, encase the capillaries. And this is what forms the blood-brain barrier. And here there are select transport routes for these different substances across the blood-brain barrier, as outlined on the right side of the screen. This is the schema showing how the periphery can impact the central nervous system, which we have all observed, and I think it's important to return to the pathophysiology of how that happens. What we see here is that there's lymphatic dysfunction that might contribute to the accumulation of waste products, which again, we see in these black stars. In the lymphatic system, we see on the top of the screen, which is a carryover from what we discussed previously. And so deficits in elimination of waste products through insufficiency of this lymphatic system can actually damage the astrocytes that support it. And this can lead to more waste accumulation, and so then a vicious cycle of damage to these cells. The lymphatic dysfunction can also affect the release of neurotransmitters, leading to deficits in acetylcholine and other types of neurotransmitters, which have been implicated in delirium, as we discussed. And then also damage to astrocytes can affect the integrity of the blood-brain barrier, which they support. And this can result in damage to the endothelial cells that make up the neurovascular unit. And so in this way, there are many systemic mechanisms for causing dysfunction of the neurovascular unit and the blood-brain barrier that are often studied in delirium. And some of these systemic complications include inflammatory states like sepsis, infection, more broadly speaking, trauma, and hypoxia. So there is significant communication between the central nervous system and the periphery. I think just to review, we see that the brain does not function in isolation. The lymphatic system aids in the clearance of waste products from the central nervous system. The neurovascular unit is important for regulating cerebral blood flow, as well as the function of the blood-brain barrier. And again, this unit is comprised of endothelial cells and these associated tight junctions that form the blood-brain barrier and the parenchymal cells, including astrocytes and neurons. So just to briefly review this communication between the central nervous system and the periphery, the brain does not function in isolation. We can see that the lymphatic system aids in the clearance of these waste products from the central nervous system. And this is really supported by these glial cells that are becoming more and more important in the study of delirium. The neurovascular unit is key for this regulation of cerebral blood flow and blood-brain barrier function. And again, this is something that's comprised of these endothelial cells, these connections between these cells called tight junctions help form the blood-brain barrier, and then parenchymal cells such as astrocytes as well as neurons. So this is really the spot where all these types of cells are working together in concert, really to try to protect the central nervous system and cause just selective influx of substances. So as a result of that, any sort of damage to these systems can result in neurologic dysfunction, including delirium. So I suspect many of you are wondering, hey, why haven't you discussed the brain yet, the brain itself? And so finally, we're going to zoom out and talk about the brain. Here is the schematic showing the different lobes of the brain. The brain is comprised of 4 lobes that have general functions, 4 lobes on each half of the brain. The frontal lobe is important for cognitive functions. You see this on the left side of the screen at the front part of the brain, important for cognitive functions and control of voluntary movements and activity. The parietal lobe in red processes sensory information, things like temperature, taste, touch, movement. And then the temporal lobe, which is in green there, this processes things like memories and also helps to integrate memories with things like taste, sound, sight, and touch. So really the area that you might think of with, you may smell flowers and this takes you back to some lovely experience that you may have had in the past. Lastly, in yellow, we have the occipital lobe. This is primarily responsible for vision. And so then all these lobes communicate with each other through networks that I will discuss in detail later. So what we experience with external stimuli is really our own perception of it. This is what we think about with our subjective awareness. And this includes auditory stimuli as well as visual stimuli and also perceptions of pain. Sometimes when these stimuli are removed, such as the neurodegenerative conditions that may result in vision loss or hearing loss or other degenerative conditions, this can result in auditory or visual hallucination, which is called Charles Bonnet syndrome. So there are a couple key tenets on the left side here when approaching this topic with delirium. A lot of the goals surround these areas, such as how do we remove unpleasant stimuli? How do we enhance pleasant stimuli? How do we control the timing of stimuli? And here on the right, I have a schema of areas of the brain that are really responsible for processing these inputs. So in yellow, we have the olfactory cortex, which is important for smell. In purple, we have the auditory cortex for processing sounds. The vestibular cortex above that is important for balance information and us understanding where we are in the world from a position standpoint. The somatosensory cortex is important for touch. The gustatory cortex is key for taste. And then lastly, the visual cortex in green, which is processing for visual information. So I think just starting with the auditory cortex, this has received significant attention, and really both in the removal of auditory stimuli for delirium purposes, such as improving sleep by removing stimuli when trying to sleep, as well as enhancing input using portable amplification devices. And then also thinking about using devices for visual aids too. So all of these things are key components of non-pharmacologic delirium prevention programs, where we see that even something as simple as earplugs can have a significant benefit when it comes to delirium reduction. Other selective inputs, such as with music, is a recent area of investigation, including actually looking at different types of music in patients in the ICU, where one randomized pilot trial looked at providing either slow tempo playlists, music that was preferred by the patient as indicated by their surrogate, as well as audiobooks, and ultimately found that there was this trend towards benefit in the slow tempo playlist group. And one theory for this is that it may have reduced sympathetic activation, cortisol, and then other things that we think about with inflammatory processes that we see are very important for neural functioning and how those relate to delirium. These all may also assist with sleep. The olfactory cortex is also an area of study in several neurodegenerative conditions, especially Parkinson's disease, which Parkinson's disease itself, as well, especially Parkinson's disease dementia, is associated with increased risk of delirium. And this is also relevant now with COVID-19, as about estimated half of people who've had a COVID-19 infection may actually experience some degree of olfactory loss. And so it's not clear what the long-term effects of this may be. And lastly, it's important to appreciate that, yes, there are these key areas for sensory processing, but they also all work together. And so the more stimulation that they get during appropriate time periods, including pleasant stimulation in terms of ways of combining all these sensory modalities, this is one of the key areas where family visitation really benefits patients for delirium prevention efforts. So next, I'm going to touch on functional networks. This is how different areas of the brain communicate with each other through these networks. There are many different types of networks that are implicated in delirium. So the default mode network is active when we're awake at rest. The frontal parietal control network, which involves the frontal and parietal regions of the brain. This is important for organizing executive control and also the function of working memory. And then the dorsal attention network, this enables attention to specific external stimuli. So one way that this can be evaluated is using EEG, and there are distinct changes in EEG frequencies, which is the idea of brain activity that's measured in hertz, and these network connectivities. One in particular that's been important in the study of delirium is the measurement of alpha frequency, which is this measurement of 8 to 13 hertz. Really, the foundation of this is going back to sleep. This is something that really supports the function of these networks. The glymphatic system is actually the most active during sleep. And so while we still don't really understand why we sleep and why we sleep so much, this really provides an opportunity for removing potential neurotoxic waste products that can accumulate while we are awake. And sleep promotion is a cornerstone of non-pharmacologic delirium prevention initiatives. And there certainly has been a great deal of interest in medication use for sleep efficacy and promotion in patients, especially those in the hospital, with the use of melatonin supplements and now orexin antagonists. And really, there could be a whole separate talk on the importance of sleep in neurodegeneration and delirium. And so I'm just briefly touching on this here. So finally, I'll touch on neurodegeneration. In the context of delirium, this is usually synonymous with cognitive impairment. This is one of the most common risk factors for delirium, and there's also a risk of acceleration of cognitive impairment following an episode of delirium. So frequently, neurodegeneration in general is associated with disordered sleep-wake cycles. And this actually leads to a cyclic pattern of continued cellular damage, where this becomes a risk factor for reduced lymphatic clearance and blood-brain barrier breakdown. But then there's also lymphatic clearance that is then reduced, and then this promotes neurodegeneration. So ultimately, this results in impaired functional network connectivity. And so this is one mechanism by which the entire circuitry of the brain can be affected. So thinking about from a top-down model and how to put things together in the context of this discussion. In terms of the different types of neurodegeneration, we do have biomarkers for some, but not all. And so that's one exciting area in neurology, which is the development of biomarkers for these different diseases that are associated with neurodegeneration. And so I'd like to go beyond Alzheimer's disease here. Oftentimes the emphasis is on Alzheimer's disease, but I think in the discussion of delirium, we need to really expand this. And so I'll just briefly touch on a couple other categories of neurodegenerative diseases that have dementia associated with them. One of the other more common subtypes is vascular dementia. And what this is so vascular dementia is the second most common type of dementia. This refers to any dementia that's primarily caused by cerebral vascular disease or impaired cerebral blood flow. Typically, this is seen in someone who may have had a stroke, and then that's followed by dementia, or maybe this is someone with some form of cognitive impairment, and brain imaging is showing that they have a lot of brain injury that could be referable to vascular disease. There are also different types of dementia that have symptoms of Parkinsonism. And so by that, I mean a syndrome of slowed movement, tremor, and increased muscle tone. These are all symptoms that you might see in your prototypical patient with Parkinson's disease. Parkinson's disease dementia develops in the context of Parkinson's disease itself. Now, this is a complication of around 10% of people with Parkinson's disease. And it's often something that happens far later in the course of the development of Parkinson's. So something you might even see a decade or more after the development of these symptoms. Often cognition is affected, and there may also be changes in personality. You may see hallucinations and delusions. And then more frequently seen in the media is dementia with Lewy body. This is distinct from Parkinson's disease dementia in that here the dementia and the Parkinsonism often co-occur within a year or so, as opposed to there being this long span of time where you may just have the Parkinsonism. Next, there's multiple system atrophy. This is a disorder that will present with Parkinsonism, but there are other features that include dysfunction of the autonomic nervous system that can result in falls. There may be ataxia and then also weakness. And then lastly, there's corticobasal degeneration. This is the type of syndrome that makes people want to study neurology in the first place. In this condition, patients may have asymmetric Parkinsonism, but also with cognitive impairment. And then importantly, these patients may have alien limb phenomenon and also some language difficulties. So to continue this thread, frontotemporal dementia is also one to think about. Here we have prominent changes in social behavior and personality, or aphasia can develop. And this is all accompanied by degeneration of the frontal and or temporal lobes. And this is something that may coincide with Parkinsonism, but is also something that's seen in people who develop ALS. There are two primary variants of frontotemporal dementia. One is the one that I think we more often think about with frontotemporal dementia, which is that behavioral variant. This is where you may see changes in personality and behavior early on in the onset of the disease course. These are differences from their premorbid personality. Behavioral changes might include disinhibition, apathy, hyperorality, compulsive behaviors. You may see people eating out of trash cans, having very disinhibited behavior. And then alternative subtypes include primary progressive aphasias. So if this is involving the temporal lobes primarily, and this is where you'll really see distinct deficits in language abilities in the different subtypes. So in summary, research is really only as good as the tools we have available. There is more to the brain than neurons. Understanding neurobiology at different levels can offer clues for studying delirium prevention as well as treatment targets, and neurologists can help with this. And we really need to understand how these complex systems work together in order to advance delirium research. So thank you for having me, and please send any questions or comments my way.
Okay, I think that we can initiate a question and answer session. My name is Alejandro Rabinstein. As Dr. Oldham said, I'm a neurointensivist at Mayo Clinic, and I want to thank Sarah for such a tour de force. It was an impressive lecture. And I want to remind the group that the question and answer tool is open. There have been some interesting questions already posted, and some of them even answered. You can look at the Q&A function, send your questions, read the previous answers. I'm going to take the prerogative of being the moderator and ask Sarah this question that bothers me so deeply. So we talk about delirium regardless of the terminology. Is delirium an entity? Is it a syndrome? Is it the final common pathway of the brain responding to a variety of insults? Because depending on whether you are a splitter or a lamper, whatever you find in research and the methodology that you use to conduct research, starting from the cohorts that you study, will have to differ. Can you illuminate me?
I think that's one of the key questions in the field, really. So, so yes, it is something that is a syndrome that we can observe. In terms of its underlying etiology, I think that there are many types of categories that we think about, and I think that's one of the reasons why I wanted to give this talk, is just to think about, well, are there ways that we can think about the risk factors that you see in the ICU when it comes to vital sign abnormalities, organ failure, but then also thinking about other types of categories we might see, such as medications that we use. And so all of these seem very distinct. And also, I think to the point I made at the beginning, which is that we're studying this in a bunch of different specialties, which is great. And we're in many ways studying these risk factors in isolation from each other, but really they're very similar. And is there a way that maybe we can go deeper than these risk factors that we're observing in the intensive care unit and in the hospital, and go even deeper into that to try to find underlying pathophysiology that might unify it a little bit more. But I think it still remains to be seen whether or not one would be a lumper or a splitter.
Yeah. And a follow-up question. So you have a patient who's sleepy, not stuporous or comatose, but very sleepy. You cannot tell whether the patient is inattentive or not. Is that patient delirious?
I think there's some debate in this. As a neurologist, with the type of exams that we're trained to perform, and I think with the way that we approach inattention, I would say that person is that person would be inattentive. But I do think that there are others who would disagree with me with that type of definition. But I know that in conversations that we've had, I think that you would agree with me in saying that person is inattentive. And certainly there are, potentially other ways that can be ascertained at the bedside. But I think just the basic answer to your question is I would say yes.
There is a question that I think is interesting, and we haven't answered it on the Q&A tool yet. And it says almost every patient develops inflammation, but not every patient develops postoperative delirium. So you have surgery. Some patients develop deliriums, others don't, even when the inflammatory cascade is activated. Why do you think some patients may be more vulnerable than others?
I think this is so important to look at. We know that some of the primary risk factors include age, and then of course underlying cognitive impairment. And some people will come to the hospital being more resilient than others. Maybe people who are younger, though we know that those that younger folks can also be at risk for delirium. I think really that the bottom line getting to this is that you do have different levels of resilience to delirium when you are exposed to a stressor like surgery. And then also there are differences in the surgery itself. So how long is the surgery? What is the depth of anesthesia? What types of anesthetics are being used? So those are also reasons why there could be some variability in the postoperative delirium that someone develops or does not develop.
Yeah, and yeah, I mean, it has to be with the external factors and the protoplasm of the patient. Both. You referred quite a bit to neurodegeneration, and I'm in the interface between neurocritical care and vascular neurology. How much do you think that underlying cerebrovascular disease that you mentioned in one of your slides and alterations in cerebral autoregulation, at least in ICU delirium, it has been noted that patients with ICU delirium have a disturbed autoregulation. How much do you think, and how much and how often do you think that those are factors?
I think that's a huge factor. Honestly, there's one person who developed delirium during my intern year that really is someone I'm going to remember forever. Was a gentleman who became delirious in the intensive care unit. And as you can imagine, as often happens in people with delirium, this person was erroneously just called delirious without there being investigation for the underlying cause. And ultimately, this gentleman was diagnosed with an MI and had to go to the cath lab. And so I think hypoperfusion is a really important cause of delirium, both acutely, but then also chronically. So then you also have just underlying vascular disease that can precipitate or be a risk factor for the development of further neurodegeneration, but then also just the potential damage to the vessels themselves that can result in issues with permeability of the blood-brain barrier and other mechanisms there. So I think that that's huge. I guess what would you say to that, since it's your subspecialty?
I think that it is a very important factor. The accumulation of cerebrovascular disease, often clinically silent until something happens. I think it diminishes cognitive reserve and diminishes cognitive resilience and puts patients at risk. But then in the setting of inflammation, you also have that cerebral autoregulation may be affected, and so to the same degree of hypotension that in other circumstances the patient would be able to tolerate, that may not be tolerated in the setting of an acute of critical illness with acute inflammation. The questions keep coming, and some are very interesting. So one of the participants asks about if you know of any animal models for studying delirium.
That's a great question. I think that does become one of the challenges in studying delirium, just because in so many other neurologic syndromes and neurologic diseases, we have been able to generate animal models, and that provides more of an opportunity to really dive into pathophysiology. So there is some potentially some rodent work that is looking at this. But for the most part, all I am aware of are mouse models, but I'd love to learn more from other scientists who are on the call.
The other question is if there are any lab tests that can be used to determine delirium, whether delirium is present at the bedside.
We know that there are these correlations with inflammation, and so there certainly is a strong body of literature that does look at the correlation between inflammatory markers and the development or presence of delirium. So I think that is one thing that can be helpful. But whether or not there is a test that says, definitively this person has delirium, that's unfortunately something that we don't have. But we do have correlations with other laboratory markers. And I think this also just gets back to this question of what is the etiology of delirium. So something, I think a framework that I find really helpful and something that I talk about with the trainees and something that was taught to me as a trainee is, this person is delirious. That's really a syndrome that you're observing. You need to get to this question of this person is delirious, and so why is this person delirious? So not just stop at this identification of a syndrome, but get to, well, why, and making sure to go that next step and asking that question. And so, while yes, there are inflammatory markers that are nicely associated with the presence of delirium, it's important to really look broadly. So maybe this person has new renal failure. Maybe this person has liver failure, and so there are a lot of other systemic lab tests that can be performed that at least can give you clues as to whether or not someone may be delirious and what that reason might be.
I have several other questions that I would like to ask, and a lot of more discussion that I would like to have. But Mark Oldham asked me to tell him when it is 3 minutes from the top of the hour, and we're at 3 minutes. So
thank you so much. Could we show the last slide? I just have 2 take-home points to make, kind of announcements, and then I'll leave it to you guys to have final comments. So the first and most important point to make is that the American Delirium Society's annual meeting will be coming up very soon, in a couple of months. There is still time to submit your abstracts. The deadline is March 6th. So basically this coming Sunday. The pre-conference that we're having will be excellent. It will be a kind of an extensive, comprehensive look at age-friendly health systems and the role that delirium plays in it. So if you're interested at all in that, please do join us. More information can be found on our website here, AmericanDeliriumSociety.org. And the second has to do with obtaining CE or CME. You can go to this abbreviated URL right here, tinyurl.com/ADS-NeuroCE to get it, or you can use the QR code. If you are watching this as a recording, you will need to go to a separate, you'll have separate instructions on claiming credit. So there was one question about, will this be recorded? The answer is yes, this is being recorded, but the recording will be available behind a paywall for ADS members. However, linking it to the other idea here, if you register for the annual meeting, ADS annual membership will be included in that. So that's just another way for those who are not currently ADS members to become a member. And then also, if you're interested in getting access to this going forward, you can. So those are my comments. I'm just going to leave this slide up here, and I'll ask you guys for any final concluding comments. We have another minute or so. My big question is, how do we get more neurologists interested in delirium? I would love to see this field grow and add neurosciences.
Believe it or not, I think that one of the problems why not more neurologists have been involved in the field is the name. I mean,
can you elaborate on that a bit more?
Well, I just don't think that that delirium was a neurology friendly term. Delirium has connotations that have turned off neurologists in the beginning, and when you talk to other neurologists who are not into the field and you say, well, I do research in delirium, they look at you a little bit weird sometimes. And also because I think that other fields have taken over, and it's very difficult to fight against establishment. In the ICU, if you try to go into causes of delirium and not just syndromic, hypoactive, hyperactive, things like that, you are blocked. So we really need to create a resistance front.
And Sarah, I'm curious. Perhaps I'll ask you that question as well. Yeah, what do you think are the barriers for neurologists in joining delirium? Is it the term? Is that something that turns kind of, I mean, I guess rubs neurologists the wrong way? Maybe they see it a little different. I'd love to get your thoughts on that.
A couple reasons for this. One being, it's possible that there are more neurologists studying delirium. They just don't call it delirium. And so it might be harder for them to be able to enter a field that where, again, we're using different terminology. So it is possible that is one of the reasons. I think that also, understandably, generally speaking, there is issues around neurophobia. And certainly as neurologists, we have been able to overcome that. But I think there is something really intimidating about studying a syndrome where at this point, we're still trying to understand what the underlying pathophysiology or pathophysiologies of it are. We don't have a specific medication for it, and that can also make doctors feel inadequate. And so I think when you're looking at other areas in neurology that are really exploding, thinking about behavioral neurology, areas in neuromuscular neurology, really all the different subfields where we're developing more and more treatments and understandings of these other diseases. I think there can be something that's really intimidating about studying delirium.
Well, I really appreciate those comments and those reflections, because I think there's a lot of work yet to be done, and I think building bridges is really important. So we're over by a couple of minutes. I really appreciate everyone for staying, those who are still here with us. This conversation could be far longer, and I really want this conversation to continue. I know we at ADS really want to facilitate this kind of conversation and build bridges. So finally, just again, thank you to our presenter and our moderator. This was just a wonderful webinar. So also thank you to those who joined us. I hope you guys have a great day. Thanks.